Welcome to the lecture series Cell Communication. I'm Dr. Janine Post of the Developmental Bioengineering Group at the University of Twente. This first lecture is on the general principles of cell communication. Illustrations in this video are based on the book Essential Cell Biology, the 5th edition, by Alberts et al. The information given in this lecture can be found in Chapter 16, Cell Signaling. In this series, I will give 5 short lectures on cell communication. This first lecture is on general principles. I will then give 3 separate lectures on the most common receptor types: ion channel coupled receptors, G-protein coupled receptors, and enzyme coupled receptors. The 5th lecture will be dealing with other regulatory principles and methods for investigating the effects of cell signaling. In this lecture, I will be talking about the general principles of cell communication and will give the general principle of feedback and signaling crosstalk. Why is cell communication so important? Well, we know that disruption in cell communication can lead to diseases, and this disruption of communication can happen due to changes at the DNA, RNA, and protein levels. If we were to identify the changes leading to the diseases, we have potential targets for intervention. And this intervention, in turn, can be a possible therapy. Cell communication occurs through a process that we call signal transduction, in which a chemical signal is recognized by specific receptors located at the cell surface. These receptors relay the signals to intracellular proteins, which results in different cellular effects. These include processes such as cell growth, proliferation, cell migration, and cell differentiation. Signal transduction is the conversion of one type of signal into another. A good analogy is the use of a cell phone. A radio signal is received by the phone and converted to sound. The signal can travel over very long distances, in this case via radio towers, from the caller to the receiver. The signal is very specific. The caller will reach the number called, and not all phones in its direct surrounding. The same is true for cells. A chemical signal is received by specialized proteins on the cell surface that we call receptors. Depending on the type of chemical signal, these signals can travel long or short distances. The signal is specific for its receptor. So, cell communication consists of 3 basic steps: reception, transduction, and response. Animal cells use extracellular signal molecules to communicate with one another in various ways. Hormones that are produced in endocrine glands are secreted into the bloodstream and are distributed widely throughout the body. Paracrine signals are released by cells into the extracellular environment and act locally. Neuronal signals are transmitted electrically along a nerve cell axon. When this electrical signal reaches the nerve terminal, it causes the release of neurotransmitters, which are then bound by receptors on adjacent target cells. In contact-dependent signaling, a cell surface-bound signal molecule binds to a receptor protein on an adjacent cell. Many of the same types of signal molecules are used for endocrine, paracrine, and neuronal signaling. The crucial differences lie in the speed and selectivity with which the signals are delivered to their targets. Table 16.1 lists many of the known signal molecules. We basically have 4 groups: hormones, such as adrenaline and cortisol; paracrine factors, such as cytokines and growth factors, for example, epidermal growth factor, depicted here; neurotransmitters, such as acetylcholine; and contact-dependent factors, such as delta. All these signaling molecules are greatly diverse in terms of chemical structure. Some are small organic hydrophobic molecules, others small or large polypeptides. Hydrophilic polypeptides bind their specific receptors on the plasma membrane, while small organic hydrophobic molecules can travel through the cell membrane to bind intracellular receptors. The same signal molecule can induce different responses in different target cells. Take, for example, acetylcholine. Acetylcholine binds similar receptor proteins on heart pacemaker cells, Ca, and salivary gland cells, in B, but with different responses in each cell type. The heart pacemaker cell fires more slowly when acetylcholine is bound, while the salivary gland cells secrete saliva. Skeletal muscle cells produce a different type of receptor for the same signal molecule and contract when acetylcholine is bound. For such a versatile molecule, acetylcholine has a fairly simple chemical structure. It is known to function in the enhancement of alertness and is necessary for being able to stay focused and for learning and memory. Every cell type displays a set of receptor proteins that enables it to respond to a specific set of extracellular signaling molecules that are produced by other cells. These signal molecules work in combination to regulate the behavior of the cell. As shown here, cells may require multiple signals to survive, additional signals to grow and divide, and still other signals to differentiate. If deprived of the necessary survival signals, most cells undergo a form of cell suicide known as apoptosis. You can read more about that in chapter 18 of Essential Cell Biology. Extracellular signals can act fast or slow. After binding of the signal molecule to its receptor, the activated receptor will relay this signal to various intracellular pathways. These pathways can quickly, in a matter of seconds to minutes, activate proteins in the cytoplasm. For example, cytoskeletal proteins. This can result in cell migration, molecule secretion, and changes in cell metabolism. Other pathway activity results in changes in gene expression and synthesis of new proteins. These processes are much longer, taking place over minutes to hours, and can, for example, result in cell differentiation, cell growth, and cell division. Ultimately, the combination of the fast and slow pathways alter the cytoplasmic machinery and thus the cell behavior. In cell signaling, we observe different mechanisms of action. Signal transduction, the transduction of a signal from outside the cell to the inside of the cell. Transformation of that signal into different cellular processes via change in activity of cellular proteins. The signal is amplified. For example, one extracellular signal can activate many intracellular molecules. And one signal can activate multiple different signaling pathways. The signal is distributed and modulated. Activity of intracellular molecules can be distributed to various processes. For example, changes in metabolism, cell shape, movement, gene expression, and many more. A signal molecule binds its receptor on the cell membrane, which relays the signal to intracellular, often membrane-bound proteins that activate many intracellular molecules, including small molecules and proteins, thus transducing and amplifying the signal. Activation of these intracellular molecules activates downstream molecules, some of which may need to integrate into protein complexes to become active. These activated protein complexes distribute the signal to various cellular responses. Activation of proteins can be amplified by positive feedback and inhibited by negative feedback loops. Some intracellular signaling proteins act as molecular switches. These proteins can be activated, or in some cases inhibited, by the addition or removal of a phosphate group. In one class of switch protein, the phosphate is added covalently by a protein kinase, which transferred the terminal phosphate group from ATP to the signaling protein. Phosphate can be removed by protein phosphatases, turning the protein off. In the other class of switch proteins, a GTP binding protein is activated when it exchanges its bound GDP for GTP, which, in a sense, adds a phosphate to the protein. The protein then switches itself off by hydrolyzing its bound GTP to GDP. Receptors on the cell surface are simultaneously bound by multiple types of signals. These activate multiple signaling pathways that interact through intracellular proteins. This way, a signaling network is formed, and the net effect of the activated network results in a specific cellular response in which multiple transcription regulators, as well as multiple target proteins, are activated. The interaction of these multiple pathways is called crosstalk. So in summary, signals from the extracellular environment tell a cell what to do. Thank you for watching this video. In the link below you can find a quiz. You can use this for both testing your own knowledge and for giving me feedback on what was explained well and what needs work. If you're interested in watching an animation of cell signal transduction, you can find the link here. In the next lecture, I will be talking about ion channel coupled receptors.